Hello students, welcome to my channel. In this class, we will learn about what is the types of DNA microarrays based on pre-synthesized DNA sequence and in situ synthesis method. What is these two methods? We'll see. There is two broadly categorized and their bases are these. Pre-synthesized DNA sequence means those probe we are using will be pre-synthesized and then placed on the solid surface. This is the one base. And second base is we are not pre-synthesized that probe. We just place them directly on solid surface. There some chemical we use and there on that solid surface we make probes. That is the in situ synthesis method. And on these bases we categorize two broad categories. First one is glass DNA microarrays. What is glass DNA microarrays? Glass DNA microarrays involve microspots. Okay, prefabricated cDNA fragment are placed on. These are the spot and prefabricated pre-synthesized probes are used in the glass microarray. cDNA fragment are fixed on glass slides. Okay, this is the pre-synthetic example of pre-synthetic DNA sequence. First DNA microarray technology was developed and this one is the first, means when we develop a microarray technique, then this time first we use this way of the microarray. And who is the pioneer on this technique is Patrick Brown from the Stanford University. What is fabric method? Is two type of method, pen tip deposition and inject deposition. Means what does means of pen tip? We have this solid surface and we see spots are there, so we place these spots with pen tip way. We are using some technique which use this way like pen tip. And another one is inject disposition spray array. Inject deposition spray array means we just spray the probe on the solid surface, like just spray and that will arrange anyhow where the spray particle gone will spread this way. And higher density oligonucleotide microarray. This one is second type. This is also known as gene chip and also known as chip. Is involved in situ synthesis of the oligonucleotide. How will do this? Most prominent example is this. In this way, this company make all these microchips. Affymetrix. The name of company is Affymetrix and this manufacturer is the name of gene chips, in which oligonucleotide are synthesized directly coated quartz surface, directly on the quartz surface with the help of chemical synthesis. And technique allow very high spot density, and here spots density are very high, hence is called high density oligonucleotide array. Okay. Gene chip technology. What is gene chip technology? In which this company, how this company make these gene chips are use Photolithographic procedure. What is this procedure? Photolithographic procedure. It combined with chemical reaction develop a combinatory chemistry. Means we quartz wafer is coated with thin layer of light sensitive compound. Means this is our solid surface which is quartz wafer, and we just cover this solid surface with a thin layer of what? A light sensitive compound. This is why it is called photolithographic. Means we fill doing a covering a like small thin film which is sensitive to the light and use direct light synthesis and use some chemical and light to synthesize that large scale of DNA sequence or probe on that particular solid surface. Hundred of thousand of oligonucleotide probe sequence on that solid surface in large scale. Array are made on glass, solid or chips. Size of chips are less than 2 centimeter. That chips or that solid surface is called glass slide or chip, and that chip size is less than 2 centimeter. Less than 2 centimeter sized chip has a whole genome, thousand of probe on that particular chip. How does DNA microarray work? We'll discuss in next class. Okay, you have any doubt, you can mention in comment section and I will try to come out with solution to overcome your doubt. Okay, bye bye, take care.